So now, once they knew that UBE1L2 was a sequence homologue of UBE1, a known E1 enzyme at the time, they wanted to see what type of protein modifiers UBE1L2 could actually bind. And to do this, they conducted an in vitro assay where they actually found that UBE1L2 bound specifically to ubiquitin protein. And so now they wanted to see whether or not they could reproduce this in vivo, specifically in human embryonic kidney cells, is what we're seeing here in figure 2C and D. So what they used was two different plasmid constructs, one with a UBE1L2 gene fused to an HA tag, and the other a ubiquitin gene fused to a flag tag. They immunoprecipitated the cell lysates with anti-HA matrix, and then they immunoblotted with either anti-HA antibody in the upper panels or anti-flag tag antibody in the lower panels. So the one lane to note here is that when they transfected the cells with both plasmids, in other words, with both HA-UBE1L2 and flag tag ubiquitin, you can see that when they immunoblotted with the anti-HA tag, we see a protein band corresponding to UBE1L2, similar to that of when only the UBE1L2 plasmid was put into the cells. But in this case, when we actually had flag ubiquitin in there, and we immunoblot for the flag tag with anti-flag antibody, we see that the ubiquitin has now co-immunoprecipitated with the UBE1L2. So this told them that indeed, in vivo, UBE1L2 and ubiquitin are bound together. Now to further this, they just wanted to see what type of binding was actually occurring between these two proteins. And what they knew was that other E1 enzymes bind ubiquitin through a thiol-ester linkage, something that can be abolished using beta-mercaptoethanol or under reducing conditions. So if we look at D now, we see the same assay as in C was carried out. In this case, when both plasmids were transfected into the cells, again here with anti-HA antibody, we see the UBE1L2 band, but we see a decreased amount of flag tag antibody showing up here, meaning that there is a decreased amount of ubiquitin that co-immunoprecipitated with UBE1L2. So this tells us that indeed UBE1L2 binds ubiquitin in vivo, but also that they are bound by a thiol-ester linkage, which was able to be reduced by the beta-mercaptoethanol, which is added here in figure D. So now that they knew that UBE1L2 could bind ubiquitin in vivo and in vitro, they wanted to see whether or not it could actually function as an E1 enzyme, and that it would be able to activate ubiquitin molecules so that it could be passed on to other enzymes and eventually ubiquitin-relate the protein substrate. So in order to do this, they conducted an in vitro assay using recombinantly expressed proteins to see whether or not, in the presence of an E2, an E3, and ubiquitin protein, would UBE1L2 be able to facilitate the ubiquitinylation of a protein substrate. So here we see SDS page gels. The first two are stained with Kumasi dye, and the third is it's done by autoradiography. And basically what is seen here in A is that hex H9 here, run in a sample by itself here in its running position, is shifted into a higher molecular weight band when in the presence of UBE1L2, an E2, ubiquitin, and hex H9, which actually duals as a E3 protein. So what this tells us is UBE1L2 is able to facilitate the ubiquitinylation of this particular hex H9 protein. In B, they're using E6AP as a substrate, and here we have its regular running position. And then when in a sample with UBE1L2 and an E2 as well as ubiquitin, and E3 being itself E6AP, we again see a shift in position of the band to a higher molecular weight. In the case of P53, we see its band running approximately right here, we see that in the presence of its E3 MDM2, UBE1L2, an E2, and ubiquitin, we see that there is an increase in fragments that are ubiquitinated. So fragments that are now migrating at higher molecular weights. So this essentially told them that indeed E1, the UBE1L2 protein was able to facilitate the ubiquitination of HECT H9, E6AP, and P53.